Hello and welcome, dear students. Our today's topic is the development of flower as a modified shoot. Generally, every plant, after going through a period of vegetative growth and in response to environmental cues, start producing flowers, either singly or in aggregation called inflorescence. A flower is defined as a determinate modified vegetative shoot meant for sexual reproduction. A typical flower has following four parts: calyx, corolla, androecium and gynoecium. The calyx forms the outermost wall in the flower. Its component parts are called as sepals, which are generally green in color. Calyx is meant to give protection to the inner parts of the flower when the flower is in bad condition. Corolla is the next wall towards the apex and is composed of units called petals. Petals are thin, soft and brightly colored and help to attract the pollinators. Inner to the corolla is the male reproductive part of the flower called androecium, which is formed of one to many stamens, each consisting of a stalk called filament and an anther at its tip. The fourth and innermost whorl is called gynoecium, which represents the female part of the flower. It consists of carpels either fused or free from each other. Each carpel has an ovary at its base, a long style and a stigma at the tip. Morphologically, a flower is a modified stem tip with compressed internodes bearing structures that are highly modified leaves. In essence, a flower develops on a modified shoot or axis from a determinate apical meristem. Plants grow through the continuous action of these meristems. During vegetative development, the shoot apical meristem produces leaves and axillary buds on its flanks, and upon perceiving the appropriate environmental cues, the shoot apical meristem gets converted to a reproductively determined inflorescence meristem. Flowering is one of the most intensively studied processes in the development of plants. Flower develops from florally determined meristems. These small portions of cells proliferate to form the floral organs, including the sterile outer organs called sepals and petals, and the inner reproductive organs called stamens and carpels. Flower transition or phase change. The transition to flowering is one of the major phase change during the life cycle of a plant. The transition must take place at a time that is favorable for fertilization and the formation of seeds, hence ensuring maximal reproductive success. To meet these needs, a plant is able to interpret important endogenous and environmental signals. Plants growing in different regions of the globe are exposed to different climatic conditions and different day lengths. In fact, they are adapted to these conditions in such a way that they exhibit alternate vegetative and flowering cycles. It means that plants respond and behave by the interaction of their inherent genetic potentiality and the environmental conditions accordingly. Plants have inbuilt all the needed signal transduction pathways to respond to environmental signals. Internal developmental changes allow plants to obtain competence to respond to external and or internal signals that trigger flower formation. Now let us analyze signals that indicate the time to flower for a plant. Shoot apical meristem has undifferentiated central dome of progenitor cells covered by a layer of epidermal cells. Signals have to come from different modes and methods to convert such potent cells to go through the developmental programs. Signals are varied such as sunlight intensity, sunlight duration, temperature and organic chemicals such as gibberellins and sucrose. Most of the environmental signals impinge on leaves and resultant downstream products have to be translocated to the same. This means that the site of perception and the site of response are separated in time and space. These signals determine whether the shoot apical meristem will produce vegetative or reproductive structures. The signals that promote or inhibit flowering can move from roots, cotyledons or leaves to the shoot apex. Once the signal is received, the shoot meristem competence determines whether the plant will respond to the signal or not. Discovery and nature of these flowering signals. In producing flowers, different kinds of plants behave differently in response to different seasons of the year. These seasonal responses in plants were studied in detail by Gassner and Garner. They studied the behavior of plants in flowering in response to temperature and length of the daylight. The temperature treatment is popularly known as vernalization, and the difference in length of the day and night in 24 hours day cycle is called photoperiodism. Vernalization. Many biennial plants and winter crops such as winter wheat require a cold period to flower. An exposure to cold that stimulates flowering is called vernalization. Different plants have different requirements for vernalization temperatures and periods. The site for the cold treatment perception in plants is shoot apical meristem. A shoot apical meristem grafted from an unvernalized plant onto one that has received cold treatment will not flower, while a shoot apical meristem grafted from a vernalized plant onto an unvernalized plant will flower. Photoperiodism. The flowering response in plants to light treatment is called photoperiodism. Garner and Allard in 1920, while working on a mutant tobacco plant, Maryland mammoth, demonstrated a remarkable relationship between effect of day length and flowering. They observed that the mutant plant flowered only in short durations of the day and failed to produce flowers in long day durations. They termed that plant as short day plants. Based on the response to different photoperiods, most of the plants are broadly grouped into three classes as short day plants, long day plants and day neutral plants. The response of a plant to the minimum duration of light period or the dark period that ultimately determines whether the plant has to continue vegetative growth or to produce flowers is called critical day period. Finer analysis of the active part of light spectrum in flower induction reveals that the red light at 660 nanometer and far red light at 730 nanometer of wavelength are most effective in inducing or inhibiting flower initiation. Role of phytochromes in flower induction. The effectiveness of red light and far red light in inducing or inhibiting the induction of flowers strongly suggests the presence of some substance that could absorb light in the said wavelengths. By absorbing the light at particular wavelength, the said substance probably undergo excitation of chromophore and the protein bound with it undergoes conformational change. This protein complex induces signal transducing activities leading to flower induction. Search for such a compound in plants resulted in the discovery of a blue-yellow colored pigment called phytochrome. This complex gets excited with the absorption of light. At red wavelengths, its protein gets activated and acts as an aspartate kinase, and it autophosphorylates itself. The pure form of phytochrome is made up of two components. One is a protein and the other is a chromophoric component and is yellow or blue in color. Now we discuss mechanism of flower development. In 1790, Goethe proposed that floral organs represent modified leaves. This idea of a common underlying mechanism has been substantially reinforced by recent findings showing that the action of a floral meristem in forming floral organs has considerable similarity to that of a shoot apical meristem in producing leaves. Nonetheless, it is also clear that there are a number of gene products operating specifically during flower development. In many cases, these products interfere with the ground state lateral organ producing machinery and modify these processes to give rise to floral tissues. Upon reaching adult phase, plants enter reproductive phase and are competent to flower. The process of floral determination is called flower evocation. Some plants flower because of intrinsic signals, while others require environmental cues like temperatures and daylights, as already discussed. Some plants have an absolute requirements for an environmental stimulus, while others respond to an environmental stimulus but will eventually flower without one as well. During development of a plant, apical meristem give rise to all of the leaves and other parts of the plant. The plant will maintain its vegetative growth pattern until the apical meristem undergoes a change. This change actually alters the identity of the meristem from a vegetative to an inflorescence meristem. The inflorescence meristem produces small leaves before it next produces floral meristem. It is the floral meristem from which the flower develops. The floral meristem undergoes a series of developmental changes that eventually give rise to the four basic structures of the flower, that is sepals, petals, stamens and carpels. Each of these structures is derived sequentially from a whorl that develops from the floral meristem. Floral determination. Determination is the stable commitment of a cell or a group of cells to a developmental pathway and involves the obligation of a vegetative meristem to become a floral meristem. The phenomena has been described and extensively characterized in Nicotiana by McDaniel and colleagues in 1992. In general, a bud is considered to be florally determined if it produces the same number of vegetative nodes prior to flowering, whether it grows in situ, grafted to a new location along the main axis of the stem, or allowed to develop in isolation. Shoot apical meristems become florally determined prior to actual flower development. Establishing the floral meristem. The floral meristem emerges as a lateral outgrowth or bulge on the periphery of the inflorescence meristem. It is at this stage that some of the first marks of floral specific gene expressions can be detected. Once the floral meristem is established, it undergoes a stereotypical pattern of growth through a series of well-defined stages. Landmark stages include the first morphological appearance of an outgrowth on the flanks of inflorescence meristem. Now, changes in shoot apical meristem. Flowers exist in a variety of arrangements on the plants, but basically they can be considered as occurring singly or clustered in inflorescence. When they occur singly, the flowers usually arise by the conversion of a vegetative shoot apical meristem into a flower meristem. In the case of inflorescences, the vegetative shoot apical meristem is converted to an inflorescence meristem, and then flowers meristem can arise directly on the inflorescence with no preceding vegetative period. Thus, these meristems go through a number of changes to become floral meristems. Cytomorphological changes in shoot apical meristem. Despite the developmental differences between single flowers and inflorescences, the changes in shoot apical meristem from vegetative to floral are remarkably similar. One of the earliest events in the transition from vegetative to floral is an increase in mitotic activity, particularly in the central zone. The distinction between the central zone and the peripheral zone is lost as mitotic activity becomes uniformly high. Associated with the increase in mitotic activity is an increase in meristem size and usually a change in shape. Phyllotaxy changes. Commonly, the phyllotaxy becomes whorl, that is more than two primordia are initiated at the same node. Sometimes the first set of organs, that is sepals, initiate sequentially, but in subsequent sets of organs, that is petals and stamens, organs of a whorl initiate simultaneously. In maize, the meristems shift from alternate to spiral phyllotaxy as it assumes inflorescence identity. Internodes elongation is minimal in flowers, resulting in the compact arrangement of organs. Organ initiation. The general sequence of events in a floral meristem is that the meristem initiates sepal primordia either sequentially or simultaneously. Then petal primordia are initiated usually simultaneously in positions that alternate with the sepals. Stamen primordia arise shortly after or simultaneously with the petals and in alternate positions to them. Finally, the carpel initiate in the center of the meristem to give rise to a pistil. As the different organs initiate, the meristems gets progressively smaller. After the carpels initiate, from the center the meristem is used up. Now we talk about different stages in the flower development. Flower development for convenience has been divided into 12 stages using a series of landmark events. Stage 1 begins with the initiation of a floral buttress on the flanks of the apical meristem. Stage 2 commences when the flower primordium becomes separate from the meristem. In stage 3, sepal primordia then arise and grow to overlie the primordium. In the fourth stage, petal and stamen primordia appear. In stage 5, they are soon enclosed by the sepals. During stage 6, petal primordia grow slowly, whereas stamen primordia enlarge more rapidly, and also carpel starts emerging. Stage 7 begins when the medial stamens become stalked. These soon develop locules in stage 8. A long stage 9 then commences with the petal primordia becoming stalked. During this stage, all organs lengthen rapidly. This includes the gynoecium, which started growth as an open-ended tube during stage 6. When the petals reach the length of the lateral stamens, stage 10 begins. Stigmatic papillae appear soon after in stage 11 and in the petals rapidly reach the height of the medial stamens in stage 12. This final stage ends when the 1 millimeter long bud is formed and start opening. Now it is clear that the whole process of the development of flower in plants is greatly influenced both by the genetics of the individual plant and by the environmental factors. The interaction of these factors affect a series of molecular and biochemical processes leading to the transition of the plant from vegetative to reproductive development. Let us analyze genetic control of flower development. For the thousands of years, people have pondered the mysteries of flower development and how these flowers ultimately give rise to fruits. Recent studies have begun to unravel these long-standing mysteries, leading to a detailed understanding of how a handful of genes interact to dictate the production of flowers and fruits. Extensive genetic analysis has led to the identification of several key regulator genes of this important process. Most of these genes encode transcription factors or other proteins involved in the regulation of transcription, hence indicating the presence of a complex gene regulatory network that regulates flower development. In order to understand the developmental sequences and the underlying genetic control in the process of flowering in higher plants, Arabidopsis thaliana has become the ideal model plant. In Arabidopsis thaliana, several transcription factors control the transition from the inflorescence meristem to the floral meristem. The specification of floral organs is a result of mutually expressed Mad-Box transcription factors that form organ-specific protein complexes. According to the ABC model of gene interaction, each of the 4 types of floral organs is specified by a unique combination of these genes. However, the transcriptional network controlling flower development is still largely unknown. For a genetic perspective, Two phenotypic changes that control vegetative and floral growth are programmed in the plants. The first genetic change involves a switch from the vegetative to the floral state. If this genetic change is not functioning properly, then flowering will not occur. The second genetic event follows the commencement of the plant to form flowers. The observation that the organs of the plant develop in a sequential manner suggests that genetic mechanism exists in which a series of genes are sequentially switched on and off. This switching is necessary for each world to obtain its final unique identity. Let us see genes involved in flower development. Arabidopsis flowers originate as small groups of undifferentiated cells called floral meristems on the flanks of inflorescence meristem. In contrast to the indeterminate shoot meristem, flowers are determinate structures that produce a definite number of organ primordia. These organ primordia reside in four concentric rings called whorls, with four sepals in the outermost whorl, followed by four petals, six stamens, and two fused carpels in the second, third, and fourth whorls respectively. About 200 years ago, the poet and naturalist Goethe suggested that flowers were modified shoots and floral organs were modified leaves. In 1991, this assertion was amazingly confirmed by Baumann et al., who showed that they could transform the sepals, petals, stamens, and carpels of Arabidopsis flowers into leaf-like organs simply by eliminating a set of floral regulatory genes such as agamus, pistillata, and apitala. Conversely, mutations in terminal flower gene convert shoot meristem into floral meristems. Careful observations of traits at the phenotypic level often give clues about how a gene might function in a genetic cascade involving other genes. There are numerous genes required for the initiation and development of flowers, and for simplicity they can be divided into distinct classes. The first class is the flowering time genes, mutations in which cause early or late flowering. In flowering plants, the transition from vegetative growth to flowering is a major developmental switch in the plant's life cycle. The timing of this transition to a flowering state is critical for the plant's reproductive success. Accordingly, most plant species have evolved systems to precisely regulate flowering time. These systems monitor both environmental cues and the developmental state of the plant. Most species go through a juvenile phase during which flowering is suppressed and then undergo a transition to an adult phase in which the plant becomes competent to flower. Genetic analysis of several species have identified genes which determine the flowering time, such as flowering locus C, CO, and FY genes. The second class specifies meristem identity and include genes such as leafy apitala 1 and cauliflower, which specify flower meristem identity, as well as terminal flower gene, which maintains inflorescence meristem identity. The transition from shoot meristem to floral meristem requires floral meristem identity genes that both specify the floral organs and cause the termination of the production of stem cells. Agamus is a floral homeotic gene required for the floral meristem termination and necessary for proper development of stamens and carpels. Agamus is necessary to prevent the conversion of floral meristem to inflorescence meristem, but is not involved in the transition from shoot to floral meristems. The agamus is turned on by the floral meristem identity gene leafy and WUS and is restricted to the center of the floral meristem or the inner two whorls. This way, floral identity and region specificity is achieved. Once the agamus is activated, it represses expression of WUS gene leading to the termination of the meristem. A third class of genes include the flower organ identity genes, which determine the fate of organ primordia and are incorporated into ABC model of the floral development. Examples of organ identity genes include apitala 1, which is involved in both meristems and organ identity, apitala 2, apitala 3, pistillata and agames. A fourth class of genes include late acting genes that control ovule development. Extensive genetic and recent molecular studies have begun to uncover the complex array of interactions among genes in this class. Genes define the commitment of flowering. As the Mendelian and molecular genetics of Arabidopsis flower development has been extensively analyzed, it has been observed by the development of series of mutants in Arabidopsis that the leafy gene is necessary for the conversion of the inflorescence meristem to a floral meristem. Similar results have been observed of floricala gene in Antirrhinium, and has been observed that the two genes also function in the commitment of floral meristem development. Floral organ identity and ABC model. Arabidopsis and Antirrhinium are valuable model systems for identifying flowering genes and underlying their interactions. The three flowering pathways, that is photoperiodic, vernalization and autonomous pathway discussed earlier, lead to an adult meristem becoming a floral meristem by either activating or repressing the inhibition of floral meristem identity genes. These genes establish the meristem as a floral meristem. Then they turn on floral organ identity genes. The floral organ identity genes then define four concentric whorls, that sepals, petals, stamens and carpels. A model properly known as ABC model of floral organ identity was proposed by Meyerowitz and Cohen. Attempts to explain the interactions of the different genes that control the process of floral organ identity. The genes are categorized on the basis of their involvement in one of the three activities A, B and C. The gene A controls sepal and petal development, gene B involved in petal and stamen development, and gene C is involved with the development of stamens and carpels. The genes responsible for the activity A are designated as apitala 2 and apitala 1. The mutation in these genes result in the development of carpels in the first flower and stamens in the second whorl. The activity B is governed by the genes apitala 3 and pistillata, whose mutation results in the development of sepals in whorl 2 rather than petals and development of carpels in whorl 3 rather than stamens. Finally, activity C is defined by the gene agames, the mutation of which results in the formation of petal in the third whorl rather than stamens. It has been concluded from the experiment that floral organ development identity is governed by these genes, and their mutations repress the expression of each other, resulting in the inverse phenotypes. This concludes our today's lecture on the topic development of flower as a modified shoot. Dear students, in this lecture we learned about the various aspects of flower development in plants, including the mechanism of shoot apical meristem differentiation, response to environmental signals, and also about the genetic control mechanism operating and regulating the process. I hope that this will help you understand the process of flower development in flowering plants. Thank you very much. Have a good day.